We'll get started. So it's our pleasure to welcome Dr. Alexey Kostikov from the McGill Neurological Institute. Alexey is a chemist, so from one of the best chemistry institutions in Russia. Would you say is the Moscow State or the St.
Petersburg State? Yes.
St. Petersburg State is the better chemistry institution in Russia, from which he got his degree, and then. Like any good Russian chemist, he got recruited to the U.S., where he did his PhD there in Georgia, and followed by a postdoc, I think, in Syracuse, and then came to Canada. We're lucky to have him. Canada's luck, again, recruiting all the brain here to Canada. So he did first a research associate, and then a faculty position at the Montreal Neuro. And he's been a huge asset for us for collaborations. He's going to talk about on the theme of brain imaging and radiopharmaceuticals, following up on the talk by Noon last. Now Alexey is going to tell us a little bit more basic chemistry, methodology development. He's been also a great collaborator for us. We have CHR grants together, we have industry collaborations together, and he's brought here people from Dr. Sheila Singh's group, and of course Dr. Fent's talk. So I think this talk is nice in a way. It will bring the institution together in more collaborative projects. So with that, Alexey, please. Thank you.
Yeah, thank you very much. Thank you very much for your nice introduction, Sam, and also thank you very much. I truly appreciate the opportunity to present here today and just to visit the place, the McMaster University, where so much groundbreaking radiochemistry research has been done over the years and is still being done. I'm most amazed by the breadth of the projects that are happening at the radiochemistry facility. So yes, my affiliations at McGill are with so my academic affiliations are with the Department of Neurology and Neurosurgery, as well as with the Department of Chemistry. I work at McConnell Brain Imaging Center, which is part of the Montreal Neurological Institute, also known as the Neuro. It was recently rebranded. And I also have to mention that I'm a paid advisor for Fusion Air Theranostics, which funded some of the work I'm going to show today. Okay, so a few words about the Neuro. If you've ever been to McGill, it is about 10 minutes up the hill from the main campus up the University Street. We have a fully equipped PET radiochemistry facility at the BIG, which was actually the first PET center in Canada. Our IBA cyclotron was the first of its kind. In fact, it was a prototype cyclotron. It's more than 30 years old now, but it's still up and running. We are looking to replace it eventually, but because we are on Mount Royal Mountain and we are right next to a football stadium, it's a bit of a challenge from the infrastructure perspective. We have the hot cells, of course. Not all of them are as brand new and shiny as the one that I'm showing here. Some of them are indeed 35 years old, but they still do the job of shielding us from radiation, so we can't complain. And we also have two PET cameras, the PET HRRT camera from Siemens, as well as the new ultra-high resolution. Sorry? We're good? Okay. The UHR camera from the Sherbrooke University, which we slowly start to use. So I wanted to share my journey at McGill in the last 16 years, where I was lucky enough to be involved in development of PET tracers and more generally radiopharmaceuticals for both the brain and for the tumor imaging. So in the first part of my talk, I will show our efforts towards development of PET tracers for some unexplored neurotargets, including receptors for neurotrophin factors such as P75 and TR, and tropomyosin receptor kinases, and melatonin receptor type 1 and type 2, as well as some of our technical innovations in labeling with carbon-11. I will also show some collaborative projects with other PIs at the Neuro I've been involved in over the years. And then in the second part of my talk, I will talk about development of PET tracers and radiotheranostics for cancer treatment that we were able to label with fluorine-18 by either silicon fluoride acceptor technology, SIFA, or including our most recent HET-SIFA platform, as well as by strain-promoted acetylene azide cycle addition, or SPAC. Okay, so here is just a brief overview of a few molecules that we were able to label for evaluate as PET tracers candidate for brain imaging. You'll notice that all of them were labeled with either carbon-11 or fluorine-18, because radiometals such as gallium-68, for example, are not are generally poorly compatible and not with requirements for brain permeable tracers. The F18 is generally preferred isotope. However, C11 has some advantages of lower radiation burden to subjects, which is relevant for longitudinal and multi-tracer studies, as well as it allows you for scanning the same subject within the same day, within 1 day, which is not feasible with fluorine-18, for example. So now I understand that I'm speaking here a week or two after Neil Vazdev, who is truly the world-renowned top-notch radiochemist when it comes for brain PET tracer development. That means on one hand that the bar is set very high, so please don't expect me to clear that bar. I'll be happy to at least to get there. On the other hand, after his talk, I'm sure I don't have to convince you about how PET can be powerful, especially as a non-invasive diagnostic modality, especially for brain imaging, where taking biopsy samples is oftentimes not an issue, almost never, not an option, almost never an option. So the first system I will talk about is the interaction between neurotrophins or neurotrophic factors, NGF, BDNF, and two others, and their corresponding receptors, which include on one side neurotrophin-specific and pro-survival tropomyosin receptor kinases, and on the other track A, track B, and track C. And on the other side, pro-apoptotic common P75 NTR receptor. So this interaction is quite complex, and I don't want to spend too much time talking about biology, which could take a whole hour, and I'm sure Professor Fanistak would do a much better job than myself talking about this biology. But generally speaking, so downregulation of TRACs or upregulation of P75 is implicated in many neurodegenerative disorders, including Alzheimer's, Parkinson's, and many other diseases. Besides neurotrophin factors, P75-NTR also interacts with amyloid beta, mediating its neurotoxicity, and which makes this receptor particularly relevant in the context of Alzheimer's disease. So this actually became the very first target I wanted to develop tracers for when I got my first independent faculty position at the Neuro. There had been very few molecules, very few ligands of P75 receptors developed at the time, with pretty much the only ones being a series of LM compounds developed by Longo and Massa back in 2006. So among those, LM11824, this molecule, was the only one feasible for labeling with carbon-11 by N-methylation at this site. So the synthesis of the cold standard and the des-methyl precursor was relatively straightforward by amidation of theophylline acetic acid and some protection-deprotection chemistry to form the secondary precursor for the radiosynthesis, so this molecule. With the precursor in hand, we performed carbon-11 labeling by N-methylation using C11-methyl triflate. So we made this tracer, and my collaborators at the Neuro at the same time confirmed that the P75 binding of the cold standard, so of this non-radioactive molecule, so that made us pretty excited about this project, because what can possibly go wrong at this point, right? Well, the blood-brain barrier, that's what. So that's apparent from this PET image in red, and this black hole you can see right here. This is where the brain is and you can see there is absolutely no uptake in the brain for this molecule. So besides that, besides the poor brain permeability, there is also very little blocking to brain slices, and the little blocking that, the little binding that we do see is non-specific. So in the hindsight, this was not very surprising because the log D of this molecule is, well, it's very polar molecule, so it's very water-soluble molecule. And for the most of the brain permeable tracers have log D values about 1.5 to 3 within that range. And that makes them brain permeable. So that at least taught us to use some brain permeability predictive software that we've used ever since, such as CNS MPO calculator, which just came out at the time, so at least we have that excuse. But now we always do the CNS MPO calculation before we embark on any tracer development project for brain imaging. So unfortunately, there aren't many other small molecule P75 ligand ligands reported, so we turned our attention to the neurotrophin receptors, the TRACs, which boast a large library of ligands because they are also implicated in some oncological diseases. So one of those ligands was this molecule, GW2580, which also has some affinity to CSF1R, another highly relevant neurotarget. The fluorine-18 labeled version of this molecule had been reported before, but because the fluorine in this molecule is in position not activated for the SNR, for the aromatic nucleophilic fluorination, it was made in 4 steps, and the quantities were never enough to evaluate it in vivo. So luckily, in around 2025, several methodologies for incorporation of F18 into non-activated rings have been reported, including spirocyclic iodinium islets, which I'm sure Neil talked about, deoxyfluorination of phenols, and the copper-mediated displacement of baronic esters. So this type of precursor. And this is the methodology that we ultimately chose. So the precursor synthesis was moderately challenging, but my postdoc at the time, Tom, who many of you know, was able to make this molecule. And we thought that the Boc protecting groups will be required for efficient F18 fluorination, and we would need to do the acid deprotection as the second step following the F18 fluorination. Now, at first, we were a little bit discouraged to see multiple radioactive species following F18 incorporation at 110 degrees. So those are the standard conditions for incorporation of F18 under this methodology. But then we realized that this could just be the partially deprotected trees, di, and mono Boc products, and we see kind of the mixture of all of these products at the same time. So indeed, upon longer incubation at 110, and especially when we cranked up the temperature to 130, all this peak merged into one, and that one corresponds to the desired product and already deprotected product, which we called FOAMPID, which is just kind of an acronym of its chemical name. And so under optimized conditions, we were able to get the tracer in 25% incorporation and close to 10% isolated radiochemical yield, which is, again, pretty good for this type of chemistry. And that allowed us to make this tracer and evaluate it in vivo. In the autoradiography on human and rat brain slices, so this is rat, this is human brain, we observed significant blocking by the cold compound and by selective TRAC ligands, but not by the CSF1R ligand BLZ945, which suggests a selective binding of the tracer to the TRAC B, but not to the CSF1R. In the micropad, the tracer does show moderate brain uptake, so compared to our previous P75NTR ligand, with a focal retention, especially in the cortex, hippocampus, and thalamus, followed by a slow washout from most of the ROIs. Self-blocking scan shows about 10% binding reduction in vivo, And while the candidate shows some brain permeability and binding specificity, the SUV in all brain regions was below 1, which is quite low, and the binding was largely non-specific. So we published this paper with Tom, who many of you know again, as the first author. But typically, this is not the candidate that you would translate to clinical imaging. So this was the end of this of the FOMPIT study. Luckily, at the same time, we were involved in collaborative project on development of another track targeting tracer, later dubbed F-track. So the principal investigator on this project is Ralph Schirmacher, who is now at the University of Alberta, and who was also my first supervisor when I came to Canada to McGill as a postdoc. So this slide shows the radiochemistry, or the radiosynthesis of the tracer, and preclinical evaluation of the tracer in non-human primates, where F-track showed decent brain permeability. So we helped here by scaling up and semi-automating the radiosynthesis for the autoradiography evaluation at the neuro. And shortly, to my knowledge at least, this became the first novel tracer synthesized by this copper-mediated fluorination technique and evaluated in Germany, which was done in Germany. So first validated in humans, which was done in Germany. And then the follow-up dosimetry study and kind of larger scale biodistribution study was delayed by the COVID. But eventually, we performed this study back in Montreal in 6 healthy subjects. So we did the full body imaging, and it not surprisingly revealed the kind of dirty little secret of all brain PET tracers, which is that they primarily accumulate in the liver and gallbladder simply because they are designed to be lipophilic in order to be brain permeable. Hence, they go, majority of them go to the liver. Luckily, the brain is so far away from the liver, so it's not like when you're doing the cancer imaging, and most of the PET scanners in neuroscience are brain dedicated, so you only see the brain anyway, so you don't even see the liver. Liver is also not the most radiation sensitive organ, so the tracer showed reasonable dosimetry. A typical scan with about 10 millicurie of F-track would result in less than 6 millisievert effective dose, which is way below the annual limit of 50 millisievert per year. So it can be reasonably used for human imaging, including multi-tracer and longitudinal studies. And this work was led by my collaborator, Professor Alexander Thiel from the Lady Davis Institute in Montreal. So next, we started a metabolite-corrected kinetic modeling of F-track using arterial input function. Briefly, for those of you who don't know, after tracer injection into the patient, we are withdrawing arterial samples, and we need anesthetist on site to install the arterial line. Then the activity of the whole blood is measured. It is then centrifuged and to separate the plasma from the red blood cells. The activity of plasma is measured again. And then the plasma proteins are precipitated with acetonitrile. And the supernatant is injected onto HPLC, equipped with a highly sensitive coincidence detector to determine the fractions of the parent compound and the metabolites. So this figure on the left shows the representative chromatograms acquired on plasma samples taken 3, 30, 60, and 90 minutes post-injection. You can see the slow emergence of the radiometabolites and disappearance of the parent compound. Luckily, those metabolites are more polar, which suggests that they are less brain permeable, so they're getting eliminated from the brain. Also, F-track is fairly metabolically stable. As you can see here on this population averaged samples taken from plasma at 90 minutes post-injection across 19 study subjects. And so you can see even 90 minutes post-injection, we have 60% of the parent compound still intact. So that's a pretty good metabolic profile for PET imaging. So here are the population averaged PET images in several time frames and the associated time activity curves, indicating the highest uptake in putamen and thalamus with peak SUVs reaching almost 2 and slowly washing out to about 1 by 90 minutes post-injection. So all in all, F-TRAC is a good first-generation tracer for PET imaging of tracks, but ideally we would have liked to see lower uptake in non-specific uptake in the cerebellum and higher differentiation between the gray and white matter regions. So the metabolite-corrected kinetic modeling was done by Professor Pablo Rozcan, who is one of the top experts in Canada in this topic. He showed that the two tissue compartment model provides a superior fit for the F-TRAC time activity curves in most ROIs than the one tissue compartment model. And the tissue to plasma ratio reaches plateau or transient equilibrium about 70 minutes post-injection for all ROIs except the white matter, which means that the 70-minute acquisition is sufficient for this tracer. And Dr. Rozcan has just submitted this the manuscript on this work. Okay, so slightly changing gears, but staying on the topic of brain tracer development, I also wanted to share our effort to make PET probes for imaging melatonin receptors type 1 and type 2. So those receptors are implicated in sleep disorders, as you would imagine, but also in mood disorders and also even in neurodegenerative conditions. There have been several attempts in the 80s and 90s to develop the PET tracers for melatonin receptors, but they all failed. But several recently developed ligands that show excellent picomolar affinity to either one or both of receptor subtypes kind of prompted us to reinvigorate those efforts. Particularly fluorinated analogs of antidepressant agomelatine bind to both receptors, while the series of UCM compounds, 765 and 1014, are selective MT2 ligands. So the former molecules lend themselves for F18 fluorination, while the latter can be labeled by C11 by O-methylation of the corresponding phenols. So that's exactly what we did with the UCM compounds. First, we synthesized the ligands according to the known published procedures, then demethylated them with boron tribromide, and finally methylated them back with methyl iodide, C11 methyl iodide. Both radiosyntheses worked quite well with radiochemical yields of around 20%, which allowed us to evaluate both tracers in wild-type rats. And initially, we were very excited to finally see very high brain permeability from our tracers. So especially the C11 UCM765 showed peak SUV values of almost 3 in some brain regions. However, the first red flag was a very fast washout from the brain, indicating that the tracer just doesn't bind to any target in the brain very efficiently. So then next we did the blocking study, and indeed we didn't see any decrease in tracer uptake using any of the competitors which reportedly bind to MT2 receptors. In fact, self-blocking with the cult molecule here resulted in the so-called reverse blocking when we saw a higher uptake upon injecting the tracer with the cult standard. So we will see the situation again in the next few slides, and I'll try to explain what we think is happening, although this remains a hypothesis. So as those MT2 selective PET tracers failed, we turned our focus to fluoroagomelatine derivatives, which bind to both type 1 and type 2 receptors. So we thought that we may lose some selectivity, but at least we increase the chances of tracers binding to either one of the melatonin receptor subtypes, 1 or 2. So for the radiosynthesis of the free 3-fluoroagomelatine, this aromatic derivative, we used again the BPIN precursor, which I showed earlier. It worked quite well, so we got the tracer in 25% radiochemical yield, which is quite high for this type of chemistry. The aliphatic SN2 fluorination of the bromoacetamido-agamelatin, or FAGAM, was less efficient, which is not surprising given that the carbon is alpha to the carbonyl. Still, we made sufficient quantities of both tracers for evaluation in vivo in rats. Unfortunately, again, we observed a very similar picture to the UCM tracers, where with incredibly high peak uptake, especially for FAGAM, where the values reached 5, so that's excellent brain permeability. But the washout was just as fast for both of these tracers. There was no discrimination between the brain regions. And again, we didn't see any decreased uptake under blocking conditions with either agamelatin or melatonin. So on the contrary, the SUVs were slightly higher in both blocking experiments. And we hypothesize that the compounds might be metabolized elsewhere, for example, by hepatic microsomal enzymes, and by injecting the cold mass, which saturate those enzymes and thus increase the brain exposure of these tracers. So I should mention that this work was led by Tom, who did all the synthesis and radiolabeling, and my PhD student Hussein, who did all the animal work and image analysis. Nevertheless, we wanted to evaluate our tracers in higher species, and we were very fortunate to collaborate with Peter Scott and Alan Brooks at the University of Michigan, who kindly agreed to inject FAGAM as our most brain permeable candidate into non-human primates. Unfortunately, despite once again a high uptake at early time frames, the tracer similarly had a very fast washout from the brain, and we didn't see any blocking under blocking conditions. So this is a DVR analysis of time activity curves that show no difference between the baseline and blocking conditions. So it is still a bit puzzling and very disappointing to see that the compounds with such high subnanomolar, picomolar affinity to the target and high brain permeability basically still show no binding in the brain. So we now think that just that the Bmax, so the expression of the melatonin receptors in the brain, is just way too low for detection by PET. Unfortunately, that could be the case. No matter what we do, we just probably cannot develop tracers for melatonin receptors. So far, this is the end of our melatonin tracer development. I also wanted to briefly show our technical improvements of labeling brain PET tracers with carbon-11. So now in F18 Volt, in the recent year, there has been a push to translate most of the radiosynthesis to disposable cassette, which rely on so which really simplify the production of tracers. You plug your syringes on the cassette or your vials, do your synthesis, and then throw away the whole thing whenever your radioactivity decays out. So, and unfortunately, the C11 chemistry is far behind. So most synthesizers still rely on stationary valves and vials, which you need to fill before the synthesis. In addition, you need to purify your tracer by PrEP-HPLC, reformulate using cartridges, and then you still have to wash your valves and vials just in order, for example, if you want to do the second batch of the second production of the tracer, or just wash the module to extend the lifetime. So we developed cassettes to produce the C11 methyl iodide from CO2 by the so-called wet method. For those of you who don't know, the CO2 is bubbled through the solution of LAH to make the C11 methanol. The methanol is then quenched with concentrated hydroiodic acid, and then you can use this methyl iodide and kind of bubble it through the solution of your precursor. Or you can convert methyl iodide into even more active methyl triflate. So we developed this cassette, which improved reliability, decreased radiation burden to personnel, and allow for rapid production production of multiple batches of tracer if needed, because you no longer need to wash the vials in between the synthesis. So we developed those cassettes for Syntomics module back in 2017, and a couple years ago this procedure was adapted for traces all-in-one module by a different group. Also, we developed a method for the radiosynthesis of amyloid tracer carbon-11 PIP, which, for those of you who don't know, was the first amyloid tracer ever developed for amyloid imaging and became arguably the most influential brain PET tracers of the century so far, well, at least in my opinion. So we used the disposable C18 cartridge as a so-called 3-in-1 entity for C11 methylation, purification, and reformulation of the tracer. So the way it works is that we apply solution of this methyl precursor on top of this cartridge. Then we pass the gaseous methyl triflate through the cartridge, and then we selectively elute the precursor first and the radioactive impurities, so they come out in 15% ethanol, and then we elute radiochemically and chemically pure product using 50% ethanol. So you can see this on these analytical HPLC chromatograms. So not only we eliminate the washing of the module, like we did before, but we also eliminate the need for the prep HPLC in this procedure. So we published this procedure again back in 2017. And also a couple of years ago, I found this publication showing our procedure being adapted for the FastLab module by another group. So again, and finally, on the PET, brain PET, I wanted to highlight my collaborative project that was led by my PhD student Hussein in collaboration with his co-PI, Professor Marco Layton at the Department of Psychiatry. So it was focused on the PET tracer for imaging metabotropic glutamate receptor type 5, or mGlu5. The tracer is called C11ABP, and the molecule is an oxime which exists in S2 regioisomers E and Z, with only the former, the E, being the nanomolar affinity mGlu5 ligand. However, the radiosynthesis provides, or at least the old radiosynthesis provided both regioisomers, which are very difficult to separate, as you can see on the analytical HPLC chromatogram of the batch produced by our old method. So Hussein first developed HPLC conditions that afford EABP in over 99% radiochemical purity. Then he used this tracer to measure whether the binding potential of ABP is sensitive to the glutamate release triggered by the administration of ethanol to the rats, and showed that indeed we see a lower binding under the challenge conditions because mGlu5 receptors, which this tracer binds to, are occupied by the cognate ligand glutamate released following ethanol administration. Okay, so now really changing gears for the second part of my talk, which is tracers for tumor imaging. So here is a brief overview of a few radiopharmaceuticals for cancer that I've been involved in development of either as a PI or as a collaborator. So you will notice that all of these molecules were labeled with F18, which, despite of the chemical challenges of its incorporation into larger bioconjugates, such as peptides, is still preferred for cancer imaging due to its favorable decay properties. So I don't think I need to spend too much time talking about radiotheranostics in this audience, but very briefly, this is a nuclear medicine technique that integrates diagnostic capabilities of PET imaging and therapeutic capability of radioligand therapy within a similar structural, similarly molecule, depending on the isotope it is labeled with. So the most common isotopes on the diagnostic side are positron emitters F18 and gallium-68, and on the RLT side we have beta emitter lutetium-177, which is the most common radiometal, followed by up-and-comers terbium-161 and the alpha emitter actinium-225. As for the targeting vectors, peptides are the most commonly used molecules because they often resemble cognate ligands of the targets overexpressed in cancers, and they can be modified by attaching the chelator for radiometals without significantly changing their properties. A traditional theranostic paradigm relies on peptides containing a single labeling site, usually a chelator, where a diagnostic and therapeutic radiometals can be swapped. So although radiolabeling is very easy in this scenario, the molecules are not identical, which may lead to their different biodistribution. Furthermore, most used metal PET isotope, gallium-68, has suboptimal production and decay properties. So for these reasons, lately, an alternative radio-twin or radio-hybrid theranostic paradigm have gained traction. This paradigm relies on two separate labeling sites, one for the diagnostic isotope, the other for the PET isotope. And for example, you can label it with radioactive lutetium and non-radioactive fluorine to make the treatment therapeutic agent, or fluorine-18 and non-radioactive lutetium to make a diagnostic PET tracer. So furthermore, they can now take advantage of superior production and decay properties of fluorine-18. The only challenge of this approach would be the incorporation of F18 into the peptide through the formation of carbon-fluorine bond, which would be pretty challenging. Luckily, several non-conventional methodologies of F18 incorporation by boron, aluminum, sulfur, or silicon chemistry have emerged in the last couple of decades. Particularly, Ralph, whom I already mentioned, developed silicon fluoride acceptors, which undergo highly efficient isotope exchange for rapid F18 labeling of peptides. So later, this turned out to be an excellent technology for development of radio twins, as exemplified by the RH-PSMA 7.3 tracer developed by Wester's group for PET imaging of PSMA in prostate cancer, and which was recently approved by the FDA under commercial name Pasluma. So this molecule contains the chelator, the CIFA for F18 labeling, the linker, and the PSMA targeting duper moiety. Okay, so phenyl CIFAs are excellent for F18 labeling due to the presence of the SIF bond, but they also have two tertiary butyl groups to shield that bond from hydrolysis in vivo, and the phenyl group for conjugation to the biologically active vectors, usually at the matter or para position. So traditional CIFAs are these giant balls of grease, which are quite terrible from the medicinal chemistry perspective. One way to offset their lipophilicity is to introduce very polar, ideally charged linkers, such as quaternary ammonium salts. So in fact, labeling this charged CIFA-choline derivative was my very first project that I worked on in Ralph's lab when I joined his group. The molecule was still too lipophilic to be used as a choline PET tracer, like fluoromethylcholine, for example. But later, this fragment was used in the design of acetate tracer targeting SSTR2, which is now in clinics for the diagnosis of neuroendocrine tumors and some other cancers, possibly outperforming traditionally used gallium dotatate. The more elegant approach to lower lipophilicity of CIFA is to replace the phenyl with a series of much more polar heteroaromatic rings, which are more common in drug design and enable diversification of the pharmacokinetic properties of the CIFA conjugates. So to illustrate my point, if you think about the benzene being the least miscible with water solvent, solvent, and then you swap one carbon for nitrogen, and now you get pyridine, which is infinitely miscible with water. So that makes a huge difference in polarity. So we embarked on this project together with our industrial sponsor, Fusion Air Theranostics, which has all the IP rights for this platform, which is dubbed HETSIFA, which stands for heterocyclic silicon fluoride acceptors. So first, we were not sure if even making these molecules is as easy as making phenyl CFAs. We also didn't know how stable SIF bond under physiological conditions, and if the F18 labeling via isotope exchange is still efficient. Finally, the carbon-silicon bond in the heterocycle could be susceptible to cleavage. Now, luckily, we were able to make over 30 diverse small molecule model compounds based on a variety of heteroaromatic scaffolds, so pyridine, fused benzo cycles such as benzothiophene, benzofurane, indole, other indoles, and most importantly, pyrazole CFAs. We found the correlation between the hydrolytic stabilities of HETSIFAs and the SciFX efficiency, and most of our compounds incorporate F18 very efficiently with radiochemical yields of over 80%. So I want to highlight the role of my collaborator from the chemistry department, Professor J.P. Lam, as well as the Montreal-based CRO Paraza Farmer, and of course Tom, who was the first author on our recent publication on this project in JMAT Chem. So pyrazole-based HETSIFAs emerged as the champions in the screen for several reasons. So first of all, they are excellent as phenyl CFAs in F18 incorporation. They have sufficient hydrolytic stability, but most importantly, they are a lot more hydrophilic than the phenyl CFAs reflected by lower measured log D values. They are also bidirectional, so we can embed them within the peptide chain or between the peptide and the chelator, further masking the lipophilicity of the diterbutyl fluorosilane substituent. So our friends at Paraza executed the synthesis of the fully conjugatable bidirectional pyrazole CFA-based amino acid, which is highlighted in red here. So it has a free carboxylic acid on one side and the FMOC protected primary amine on the other, so it can be incorporated into a peptide chain by the solid phase synthesis. So we made a series of SSTR targeting HETSIFA-8 conjugates containing metal chelator for potentially imaging or treatment of neuroendocrine tumors. And the entire synthesis was done using SPPS. The conjugate can be directly labeled with lutetium-177 to deliver a potential radiotherapeutic, or we can chelate non-reductive lutetium and label this molecule with fluorine-18 to make identical radiotwin. The measured log D value of the conjugate was much lower than the SI-8, which is currently in clinical trials, and F18 labeling works excellent, so as we were getting about 75% incorporation in low activity optimization, and our conditions were scalable for high activity labeling with over 2 curies of F18 yielding more than a curie of the labeled peptide in over 50% radiochemical yield. So this synthesis can be done using either manipulator arms or or it can be also automated. So for the high activity labeling, the biodistribution and PET imaging study, I can't thank enough Sam and his team, Rafael, Ayman, Mohammed, Afaf, sorry if I forget anyone. Big thanks for all of you for helping us to evaluate our tracers. So in wild-type mice, the tracers showed a very favorable biodistribution with the majority of radiotracer found in the urine and bladder, suggesting rapid renal excretion. PET image was consistent with the ex vivo biodistribution, as you see most of the activity in the kidneys and the bladder 90 minutes post-injection. Now we are well aware of the fact that there are plenty of TATE imaging and therapeutic agent on the market, and it would be difficult for us to kind of enter this market and compete. So that's why that was the end of our proof of concept study, using TATE just as a well-established benchmark. So instead, we focused our efforts on development of theranostic radio twins targeting gastrin-releasing peptide receptor, or GRPR. This receptor is overexpressed in prostate, breast, and other types of cancers, representing alternative theranostic target for PSMA-negative tumors, which can be treated with PSMA targeting Posluma, PSMA 617, INT, and others. So the two known peptides targeting GRPR are NeoB and RM2, which differ only by the kind of C-terminus and the linker between the peptide and the chelator. So both peptides have been labeled with gallium-68 and lutetium-177, and the corresponding theranostics are in clinical trials for diagnostic and treatment of prostate, here on the left, and breast cancers. However, no F18 labeled GRPR targeting peptide has been approved so far, nor a successful radio twin has been developed. So we synthesized a series of GRPR targeting radio twins, which consist of the following building blocks. So either RM2 or NeoB binding domain, our het CFAs for F18 labeling, the variety of simple linkers, and chelators for radiometals. Once again, all peptide was synthesized using standard SPPS protocol, followed by the global deprotection and cleavage of the resin. So we've by now synthesized over 30 peptides and measured their affinity to GRPR in vitro, again thanks to Ayman. This slide shows only the affinities on both Ki scale at the bottom, on top, and the pKi scale pKi at the bottom and the Ki on top. So successful imaging agents should have affinity in the single nanomolar range, so somewhere in this green area, like RM2 and NeoB. So on this plot, you can see that most of our tracers retain affinity, and two of them had Ki's around 10 nanomolar affinity, and by now we have new conjugates which are which have even higher affinity, very in low single-digit nanomolar range, comparable to NeoB and RM2, so firmly here in the green zone. So although we think we have a better candidate by now, FTX-279 has been the most fully evaluated radio hybrid so far. The empty chelator was radiolabeled with lutetium-177, pretty much quantitatively, which allowed to make several millicuries of the lutetium-labeled tracer for the longer time biodistribution study. Also we can label non-radioactive FTX-279 with the natural lutetium in it with the fluorine-18 and evaluated by PET imaging. So again, the F18 labeling works incredibly well. We get more than 50% radiochemical incorporation and high molar activity, high radiochemical purity, so we can evaluate them in vitro. And again, I must acknowledge the wonderful work done by Sam's team, particularly by Mohammed and Afaf labeling these molecules. The biodistribution study of F18 labeled FTX-279 shows beautiful uptake in the GRPR expressing tumor in PC3 mice model. So as you can see here by the blue bar on this plot, more importantly even, the uptake was fully blockable by the NeoB, as shown by the adjacent green bar. So that shows that uptake is selective to GRPR. Uptake in non-targeted organs, including kidneys, liver, and others, is very low, and bones. The only other organ that shows high uptake is the pancreas, but that is normal, so all GRPR targeting theranostics show uptake in pancreas. So finally, we keep saying that the F-18 and lutetium-177 radio twins should have similar biodistribution, but I don't know if anyone has ever actually shown it. And we actually did, so we showed that the biodistribution of F-18 labeled and lutetium labeled compounds have identical biodistribution in all organs, but particularly relevant is the tumor. Overall BioD of FTX-279 is comparable or even better than that of gallium-68 Neo-B, which has been advanced by Novartis. So PET images acquired using F18 FTX-279 are in good agreement with the BioD data. So we see high and fully blockable uptake in the tumor. Most of the rest of the activity is in the bladder, and some still remain in the kidneys, but this activity quickly washes out from the kidneys, as we know from the biodistribution experiment at longer time points. So here are some of the time activity curves. So you can see rapid activity uptake in the tumor, which plateaus by about 1 hour post-injection. On the contrary, the washout from most non-targeted organs is fast, except for the pancreas, where the uptake is specific. And critically, bone uptake also remains at the same level throughout this time frame, indicating that the tracer does not undergo defluorination. So the tumor retention at longer time point is not an issue for the PET tracer, of course, but it's very important for the radioligand therapy, where you want the radiopharmaceuticals to stick around longer to deliver its cytotoxic radiation. At the same time, we really don't want to see retention at longer time points in non-targeted organs to avoid unnecessary damage. So the tumor of retention of lutetium-labeled FTX-279 is down to about 1.5 ID per gram, which is a bit lower than we hoped for. We hoped for about 5 ID per gram. But on the positive side, the retention in all non-targeted tissues is pretty negligible at this point. Again, must acknowledge Raphael for all this beautiful, amazing animal work shown on all of my slides. And another advantage of the radiotwin theranostic paradigm is that we can chelate non-radioactive isotopes of different radiometals used in nuclear medicine, label them with F18, and study their biodistribution without access to those expensive radiometals and sometimes poorly available radiometals. So among those, terbium-161 is the most promising one. It promises to perform even better than lutetium for radiotherapy. So we also labeled our compounds with terbium, and at the same time, by modifying the linker, we managed to increase the hydrolytic stability of the SIF bond in aqueous media and further lower its log D values. So now we think we have even better candidates. So by incorporating terbium, which is a little bit surprising, we have even better affinity to the target than with the lutetium analogs. So we have very high hopes for our tracer candidates and hope to advance them to human imaging very soon. This translation is being led by Chris Waldman, who is the Chief Scientific Officer of our industrial sponsor, Fusion Air Theranostics. And do I have more time? Or should I wrap it up? I have like maybe 5 more minutes worth of slides. Good? Okay, thank you. So I want to talk about slightly different approach to peptide labeling that we explored a while ago, and that is F18 fluorination by click chemistry. So if you look at the challenges and requirements for radiochemical reaction to make radiopharmaceuticals, so they must be fast, first of all, as we raced against the decay. They must be high yielding and selective to maximize the radiochemical yield, and modular to be applicable to radiosynthesis of multiple tracers. The products must be stable, including in vivo metabolic stability, And ideally, the reaction conditions must be biocompatible and proceed in non-toxic solvent to avoid losing time to reformulation of those tracers. Now, if you look at the requirements to click chemistry reaction from the seminal publication in Angewandte Chemie by Sharpless, almost quarter century ago now, you will see that there is a lot of overlap between between the requirements. You can even tell that this is the match made in heaven. And indeed, can I lost my? Shortly after the original report on copper-catalyzed azide alkyne cycle addition, so commonly called the click chemistry these days, radiochemists quickly adapted this methodology for labeling of peptides. Both F18 azides, so here is F18 labeled azide, and F18 labeled alkynes were developed by 2007, and shortly after many other click synthos have been reported. Now, copper click satisfies most of the requirement I mentioned, so it's very fast, it's high yielding, of course, modular, selective, but the only issue here is the presence of the cytotoxic copper ions, which means that this reaction is not biocompatible. So, which means that, for example, it's not applicable for pre-targeting approach. The radiosynthesis requires purification and extensive quality control procedures to make sure that we purify all the copper ions. Furthermore, this chemistry is not applicable for labeling with copper-64 and other isotopes of copper, which found their application in radiopharmaceutical research. So we focused our attention on the copper-free, or also known as strain-promoted acetylene azide cycle addition, SPAG, which was originally reported by Bertozzi. This click reaction proceeds without the copper catalyst because the driving force here is the release of strain of the triple bond, which of course wants to be planar as any triple bond, but now here in this molecule it is strained within this cyclooctane ring. So that decreases the activation energy for the cycle addition, which now proceeds at room temperature in absence of any catalyst. So the tosylate precursor of ADIBO, which stands for aza-dibenzocyclooctane, so this molecule was made by the group of Professor Popik at the University of Georgia, who was actually previously my PhD supervisor in the University of Georgia. So he sent us some of this precursor, and at McGill, we developed the conditions for fluorination by standard SN2 to make F18 ADIBO, and later they provided us with even more strained ODIBO, or oxo-dibenzocyclooctane, which we labeled using similar conditions. And later actually found that the ADIBO tosylate and the cold standard have become commercially available by this company. Like, I don't get any royalties from this company, so I don't have any conflicts of interest here. All right, and yeah, so we published both of our work, both of these projects, and I'm particularly proud of the artistic work that we did for the journal cover for one of these publications. All right, so but does this radiochemistry work for peptide labeling, you might ask? Well, it does, and it does so very efficiently. So when we mix the ethanol solution of ADIBO with the TET peptide modified with the azide on the terminus, we see and we monitor this reaction by analytical HPLC, we see a very rapid disappearance of the cyclooctane and the formation of our click triazole product. So this compound. See, almost complete disappearance of the ADIBO and the formation of this guy. On the UV trace, you can also see the click product and the starting material, but no other byproducts, which indicates that chemistry proceeds very cleanly. So it also works with F18 ODIBO, which I won't show the HPLC traces for because they look very similar. And that ODIBO click is actually even more efficient because, so you can see it on this slide, because of the higher strain provided by the oxygen in the ring compared to the nitrogen. So this chemistry is even more efficient And you can see this here showing radiochemical conversion of ADIBO in orange versus ADIBO in green versus ODIBO in orange at equal concentration of peptide. So you can see in more dilute conditions, you really see the difference between the two. Okay, so finally, conclusions and reflections from my journey. So development of novel PET tracer is extremely challenging. I mean, thank you, Captain Obvious. As it can fail at so many junctures, including synthesis of precursor. We had these situations too. I just didn't show them because they're not very exciting. There's not much to show. Radiochemistry can fail. Also, I didn't show these examples because this is not very exciting. But then also in vitro, and in vivo, your compound can still fail. However, it is incredibly rewarding when the tracer or methodology actually works and is translated to clinical trials. Again, sorry for stating the obvious. I was fortunate to contribute to the development of several tracers, including SI-TATE and F-TRAC, developed mostly by Ralf Schirmacher, that are extensively being used to image human diseases. We developed several methodologies for C11 labeling and F18 labeling. Het-CIFA-based GRPR targeting radio twin theranostics are our most promising compounds right now, as they show very encouraging results in mice and are being translated for human imaging. And also next generation track targeting tracers are being developed in collaboration with McMaster University. And finally, I would like to acknowledge everyone who was involved in this work. First of all, the team, especially Tom and Hussein, who are involved in most of my projects. My collaborators, so my former supervisors, Ralph and Vladimir, and my most active current supervisors, including JP, Sam, Chris, and collaborators from the Neuro, Alex Steele, Pablo Ruchan, Mark Layton, and many, many others. I also want to thank the team at the PET cyclotron radiochemistry facility at the Neuro, who provides us with the access to the radioisotopes, as well as the funding from CIHR, NSERC, Fusion Air, and funding from my institutions. And I also want to thank you for your attention, and I would be happy to answer your questions. Carbon-11, yes.
You can. But you know, it's amazing. You start with the cyclotron. You create carbon-11 with CO2. You then make, you make methyl iodide, and then you make methyl triflate, and then you attach that and new HPLC purification. We still get to do that for human imaging. That's a feat of, I would say, organization, dedication, and
which means that, yeah, you also have to do the QC before that.
Yeah. Quality control, that's right. And you saw, that actually brings me to one of the questions. How do you, with some of the earlier tracers you were showing for brain imaging, you could do methylation of amines, but you chose to do F18, for example, for your tracer development. Is there, how would you choose which path to take if you have both options of carbon-11 development or F18 development?
I mean, all things being equal, I would always choose F18 because it gives you more flexibility to work with, and also fluorine. When you have your fluoride, it's an aqueous solution. All of your carbon-11 synthons are gases, so you have CO2, methyl iodide. So I would say it's a more complicated chemistry with C11, but sometimes you don't have a choice. Sometimes you don't have fluorine in the atom, and you have this this methyl dangling on oxygen and nitrogen, and you look at it, it's like, yeah, well, it just begs to be, this molecule begs to be radio-labeled with C11. And yeah, if it's
easier to start, if you're like
yes.
renal and you have capacity to do brain imaging right away, just to see that and put in the patient. If it works, then you maybe develop an
yeah.
version. Questions?
Alexey, really nice work.
Thank you.
You showed those melatonin binding molecules, and you said that they get cleared really fast.
Yes.
Fast, right?
Yes.
So would multivalent binding be helpful there if you, say, stuck 2 of them together or 4 of them together? Would that, do you think, help with retention long enough
That's an interesting idea, but also those are small molecules. So those are not like peptidic fragments that bind to the target. Those are really small molecules that just find their way into the binding pocket. So I'm afraid if you like put it, for example, on the dendrimer, I think it will have a hard time of getting into this binding pocket and getting into the brain too. So I think it would be, for this particular application, that would be more challenging. That's my gut feeling, but I could be wrong. Maybe it would work. Yeah, but then it becomes a large molecule, and usually in brain, so in order for a molecule to be brain permeable, most of the time you need the molecular weight to be below 500 Daltons. So if you try to kind of stick it together, then you're quickly exceeding this limit.
Alex, I was looking at the data that Alexey had, and he kept alluding to it, but never said it. If you guys look at Neil's pictures and some of what put us on the Nature at McMaster with the DOPA imaging, you sometimes forget that fluorodopa, if you just inject it, it's going to have the similar type of fate that Alexey showed here. It gets decarboxylated and never gets into the brain. So when we do brain imaging with dopa, in fact, we saturate the decarboxylase enzyme with the addition of carbidopa. So is that something that you thought about? Maybe the fact that you added some more cold compound instead of blocking, you saw more of the compound getting into the brain. You're probably, as you alluded to, stopping some kind of an enzymatic reaction that's creating these metabolites. Very similar to what we see with dopa. We add an inactive compound like carbidopa, saturate those enzymes, leaves the intact dopa to get into the brain. So is that
that's a great idea, but then how would you prove? So you see increased uptake, I guess, but then how would you prove that it actually binds to the receptor?
We have all the solutions. You're already doing all the metabolite analysis in the brain of a mouse. So if you can show that actually what you have in the brain, take the brain, homogenize it, show that you're seeing metabolites, and show that you see less metabolites with even addition of your cold compounds, then you know that's some kind of an enzymatic reaction. Good stuff. Very similar to the dopa story. So
yeah, that's true.
The way we do some of, but you were saying all the answers in your presentation. Really nice work, though.
Thank you.
Amazing, Alex. I'm really looking forward to our next story. I mean, that story with the GRPR, those compounds that you're showing, those are all, multi-tens of millions of dollars that are being shown, and the compounds that we have developed with the CEPA chemistry are better. So I think that's one, and then we're going to work on other ones for brain imaging. So looking forward to our next presentation and next set of data.
Looking forward to it too.